You can, Kristin, right?
Yeah, I can.
Great. So it's a pleasure to welcome Dr. Kristin Swanson. So Kristin and I have been friends and colleagues for many years now, probably at least 2 decades, which makes me feel old.
Oh yeah.
So Kristin has been a pioneer in many ways, but I'll come back to that once I give a brief bio. So she got her bachelor's in maths in '96 from Chilean University. And then she got a master's and a PhD in mathematical biology at University of Washington, where she was one of Jim Murray's students. And that's really where she got interested in cancer modeling, specifically in brain cancer. She did a brief postdoc at UCSF and then went back to the University of Washington to have a faculty position in 2000. And importantly, that position was in both neuropathology and applied math. So she saw early on about the importance of really being close to the data. And in fact, one of the kind of pioneering aspects of Kristin's work is that not only did she see the importance of the data, but she started collecting the data herself. And that started way back in 2000, when most of us were just hoping one day we'd get to access some data. She's currently a professor and vice chair of research at the Department of Neurological Surgery at the Mayo Clinic in Arizona. And she's the co-director of the Precision Neurotherapeutics program and director of the Mathematical Neuro-Oncology Lab at the Mayo Clinic. She's got many papers, over 120, and even more abstracts, and really has been sort of a leading light in the field of mathematical oncology, especially one that has potential for clinical translation, working directly with oncologists to help facilitate decisions. So it's really a pleasure to introduce Kristin to give us our talk today on sex, drugs, and radiomics of brain cancer. So Kristin, take it away. Love that title.
Thanks, Andy. I'm excited to be here, guys, and tell you a little bit about some of the work that we've been doing. And I thought, the IMO folks would be most interested in hearing about newer stuff. So I'm not going to focus much on stuff that you probably might know more, but more stuff that's been happening over the last couple of years in my lab. And it all focuses on sex, drugs, and radiomics. So I'll tell you all about all of those. Thank you again for the invite, Sandy and David. All right, so see if I can make my slides move. There we go. So the type of brain cancer that I work on mostly is gliomas. And gliomas are primary brain tumors that develop, meaning that they develop in the brain and from brain cells. So the most aggressive of those are the glioblastomas, and they're particularly deadly. People often use the term uniformly fatal disease, but it's not only a cancer, but it's a neurological disease. And I'll explain a little bit more about that in a few slides. There are about 13,000 new cases a year ish. Certainly in the US, that's about the right number. And the standard treatment is we throw everything in our arsenal at these patients. We give them the maximum possible surgery. So largest surgery with the least cognitive and functional deficits, followed by radiation therapy, high dose radiation therapy, concurrent with temozolomide, an alkylating agent chemotherapy, followed by adjuvant chemotherapy until progression. And progression, like I said, it's a uniformly fatal disease. So it's considered a uniformly fatal disease. So there's often fairly early progression with a median survival of, something like 16 months and a 5-year survival of, in the more optimistic cohorts of patients, maybe 5%. With really no change in the last 20 years. And one could really argue very little change over a long period of time. Really, I think only 4 drugs and one device have been FDA approved in the last 30 years. And basically, as that implies, most all clinical trials fail. And why is that? So let's click through. So why is that? So one of the reasons that this disease is particularly bad is the fact that every patient is so wildly different. So glioblastoma was previously referred to as glioblastoma multiforme. Multiforme referring to the fact that it's heterogeneous nature from what you look at under the microscope, but also the heterogeneous nature across patients. So to give a sense of that, here are 15 patients all receiving the standard of care. In this case, they were in locations so they couldn't have a large surgery. So you only have a biopsy, but if you scale the tumor size to 1 at diagnosis at the day before radiation and chemotherapy start, you see that during radiation and chemotherapy, there's a wide difference and wide variety of responsiveness, some not responding, presumably. Presumably this blue patient is not particularly responding very well to that therapy, whereas this orange patient is receiving a huge bang for their buck from that standard of care therapy of concurrent radiation therapy and chemotherapy, followed by adjuvant chemotherapy. And you see that there's long tails of robust responsiveness in a small subset of patients, but there's also a ton of patients that are not getting that bang for their buck. And so what this really pushes for is a personalized medicine strategy for this disease. But to personalize, one has to know what's going on in each of these individual patients. And so you need predictive models, and that's where the maths comes in. So to do that, we really have to drill down on what we know about the disease. And what we know about the disease is really informed by imaging. So MRI gives us insights into the disease because it's in the brain. So if there was a tumor, let's say in your leg, sort of like a leg sarcoma, you could imagine sampling that tumor fairly often, being able to collect tissue over time as you're receiving different treatments and, collecting, serial biopsies. But in the brain, neurosurgery is one of those things that you do not do very often, right? So a typical patient may have brain surgery 2, maybe 3 times during their lifespan with glioblastoma. So much of the insights that you have to get for this disease come from imaging. And so here's a reconstruction of a patient's MRI, and in red is the outline of the edge of what's abnormal on MRI for a certain type of MRI. In this case, it was gadolinium-enhanced T1 weighted MRI for this individual patient. And this patient, when you zoom in, if you could miraculously zoom in to a single voxel level for this patient's tumor, you would see something like this. Over a 24-hour window, these cells are migrating and proliferating at the leading edge of the tumor and really within the tumor. So this is what makes this disease particularly unique. And this what makes this disease not only a cancer, but also a neurological disease, because where in the black area in this movie here is normal brain. So those are neurons and other normal brain cells being interacting with these tumor cells to a degree that is unusual for other cancers, right? You would think of other cancers as sort of a pushing phenomena. In this situation, it's much more of an intercalation between the tumor cells and the brain. Okay, so in this video, this is from an animal rat brain slice assay video microscopy, but this gives you a sense of how fast these cells are migrating in 24 hours, and the rate at which these cells are migrating is exactly the median rate that we observe for human data as well. So as a result of that, we published over the last several, forever ago, started publishing on a series of models that were in the reaction diffusion context, right? I can't be a student of Jim Murray without focusing on, at some level, on reaction diffusion models. But reaction diffusion models were actually a really good fit for this sort of model, for this sort of disease, because of the diffuse invasion that you see there. So diffusion was a very nice model for this, and we will refer back to this a little bit in the rest of the talk, but I won't focus much on this series of work. The one thing I do want you to take from this is the fact that the dispersal coefficient for these tumor cells is quantified as D, and rho is the net proliferation of the rate of these tumor cells. And so you'll be able to refer back to those characteristics later on. So what this really means is that using this sort of model and tuning it to each of the individual patients, you can then connect what you see on MRI, which is the top part of the image here, with the diffuse invasion, this diffuse intercalation of individual tumor cells into the periphery around that abnormality on MRI, and to be able to connect those dots. And so you can see how quickly it becomes apparent that the degree of this invasion becomes important in terms of understanding the heterogeneity of response across individual patients. So this is the backdrop of the disease that is profound. And one of the most necessary things that Sandy commented on was being able to collect a robust cohort of patient data to analyze these sort of patterns in individual patients. So starting 20 years ago, I started a IRB protocol, IRB approved protocol that allowed me to consent and collect patient information, including relational, including data on therapies, medical records, imaging, what images there are. And then we built a suite of tools around those images to build segmentation tools, how to be able to extract what's abnormal on the MRI, and that's what we're illustrating on the bottom. And so basically each patient in the database has this, these curves over time, which represent the MRI size over time. And these are the different types of MRI you have here about gadolinium enhanced T1 weighted MRI is the one I showed you before, and where we saw that nice red segmentation. And the T2 and weighted and flare images are other images that we see that are very routinely used. There are other kinds of multi-parametric MRI that are used clinically, but these are three very common ones that basically every patient will get at some point during their disease progression. But the point here is that we've built a large database that, again, has over 3,000 patients with over 70,000 MRIs that have for patients that have received over 140 different treatments. And so now that gives us, from like a data science math perspective, it gives us a lot of potential ways to explore the biological heterogeneity and responsive heterogeneity across patients. So the first axis of heterogeneity that you would think in the world of personalized medicine we would know something about is sex differences. And the reality is, up until about a year ago, nobody was, well, not quite a year ago, maybe 4 or 5 years ago, for the very last few years has been the first real acknowledgement that there are significant sex differences in this disease, despite the fact that it's long been known that there's a skew in terms of the abundance of the disease, with males more likely to have the disease than females. But there is, especially in the case of the current standard of care, where we have radio chemotherapy with temozolomide followed by adjuvant chemotherapy, there's an increasing realization that there is a preferential benefit to women in that, in terms of outcomes from that treatment. So to explore this, a recent paper that we worked on with colleagues of mine at Washington University, Josh Rubin, he's a pediatric neuro-oncologist who has a ton of interest in and this really been a pioneer in understanding sex differences in brain biology and brain cancer biology in particular. We began working with him on some work that was looking at molecular pathways and phenotypes of female tumors versus male tumors and trying to differentiate those. And so foundational to that analysis is the idea of this jive analysis. So it's a statistical approach where you can separate out the overall omics signal. In this case, it's transcriptomics. You find out what's common between the male and female cohorts. You subtract out what's common, that's the joint, and then you separate out a pile for male-specific and female-specific unique expression patterns, right? And so when you do that, this is the sort of pictures that you get, and then you can generate clusters. Basically, you can do a cluster analysis on the female specific, and you can do a clustering analysis on the male specific, and simply look what's different, right? So at a high level, here's the key. There's about 116 overlapping genes, patterns in this analysis, but there's a significant amount of sex-specific patterns. And it turns out those patterns matter. From a biological perspective. I guess one other thing I'll mention on this slide is one of the simplest ways you can analyze sort of this sort of data is to, once you do a female specific and you get like 5 clusters, you see them annotated here, 5 clusters, and you do a male specific, you get 5 different clusters. Then you look at the clusters and it's kind of, a logical first step to look at those clusters is to go, okay, well, let's look at the 5 female clusters and say which one is, has which cohort of patients has the longest survival. And in the female cohort, the signaling that drove that or connected to the longest living cohort were patients with that expressed a lot of integrin signaling. And so there's some hypotheses about what that means. And on the male side, the clusters that were most strongly associated with long survival were patients that expressed cell cycle control mechanisms, so controlling of proliferation. So there's clearly 2 different ways in which tumors can lead to long survival in between the sexes. So that's the first pass. But again, circling back to the sort of rich data that we routinely have in this disease, right? You may have one sample where you might be able to get RNA-seq, but you're going to have serial MRI for these patients. We started looking at the connection across multi-modal MRI and appearance and the connection between these different types of MRI between males and females. And in blue here, we scatter the correlation of the different sort of standard MRI sequences of the signal for that sequence versus that same, sort of correlogram across these various sequences. And just grossly looking at that picture, you go, okay, blue and red seem to be similar, but definitely there's potential for difference there. And you can even try and build sort of logistic regression models to be able to differentiate and predict which tumors are male versus female from those imaging signals. Turns out you can, and you can differentiate these things out. Even over and above that, you recall that we were talking about segmenting an MRI and being able to outline what's abnormal on the MRI. You can use deep convolutional neural nets to design a automatic segmentation so that you take an MRI and produce the red abnormality, the estimated red abnormality. Turns out if you train a model for males versus females, you get different answers if you use the male model on female data and the female model on male data, which further supports that there are some intrinsic MRI differences between the sexes. So are there differences in the mapping between what the MRI is doing? So I think we've clearly shown that the MRI imaging features are different, but is the tumor biology underlying those imaging features also different? And that's what we've sort of alluded to with that RNA-seq work. And the first simplest way you can do that is to look at the invading edge. So in this bright region here, kind of outlined with my cursor, is what the extent of the gadolinium-enhanced T1 weighted MRI is. And that's classically associated with the tumor. And that is the region that is segmented or resected during surgical resection. And this penumbra around it, that is hyperintense on T2 weighted MRI, is thought to include invaded tumor cells and associated edema or swelling in the brain. So if you biopsy in these regions of the T2 abnormality, so outside the T1 GAD where you expect to run into frank tumor, but out here where you expect to run into a few tumor cells, you're just not sure how many, as I was illustrating on the previous modeling simulations, you find that there are way more tumor cells in men than women in these regions. So now we're getting even more complicated. So what you see on imaging might be bright, similarly bright for a male and a female tumor, but they mean something different in terms of, at least in this case, the diffuse invasion of those tumor cells. Further, if you take cohort, one of the things that we've been particularly interested in over the last several years has been collecting image localized biopsies. So during surgery, you capture on the neuronavigational system where you're at surgically, so you can record the location of that specimen, that biopsy that you collect. And so you have all these different MRI sequences, and you can use machine learning and other more mechanistic modeling approaches to connect the biology you reveal on this piece of tissue to what you see on these different MRI sequences at that location. So it's maybe bright on gadolinium enhanced, but it could be bright on T2, but it could be dark on RCBB, or some various combinations of this heterogeneity that you see. When you do that, you could also sort of think about how are these different imaging sequences connected to each other? Like generally speaking, if it's dark here, you should expect some sort of brightness on the T2, for example. There's sort of intuitive expectations about the biophysics of MRI and how MRI changes on one sequence should manifest in terms of changes on other sequences. But what's notable is if you just do a simple correlogram, in this case, it's represented as a chord diagram between the different imaging sequences that we might collect for our patients, you see here's the way they're correlated for females, and there's the way they're correlated for males. They're not correlated the same. So again, further substantiating that there is a connection difference between these sequences. So we're building the story about how sex differences might underlie not only the basic biology, but also the imaging features. But how does all of that stitch together to impact patient outcomes? So again, we kind of got excited about chord diagrams. So here's another kind of chord diagram, right? Which is looking at this is called a Circos plot. And we're on the top, you're connecting, this is overall survival for the cohort. In red is female and in male is blue. And you're either connecting overall survival, and you see overall survival for females is connected to how diffusely invasive the tumor looks, that D coefficient from the standard models that I was showing you very early on, the reaction diffusion model, or how the age and age at diagnosis, which age is the most like #1 predictor for outcome for this disease across sexes, and so that's consistent. Overall survival from the males is age at diagnosis and T1 GAD radius, the size of T1 GAD on MRI, so how big the tumor is. So size matters to men, but invasion matters to women. And then same thing for short-term survival, you have differential patterns with 4 different aspects of the imaging features being connected to short-term survival, where only there's 2 in males, where there's only 2 in females. So again, grossly from this picture, one should just take away that the way the imaging features change matters differently between the sexes. So I hope that this is a compelling case for sex differences across this disease. So one of those things we referred to in the previous slide was the fact that diffuse invasion had a significant difference between the diseases. And this may be related to that integrin signaling story that I mentioned earlier. But it is notable here, for instance, in a pretty large cohort of patients, that the more invasive the tumor is, matters a lot for women, but does not matter as much for men. And again, invasion as captured by the degree to which there's an extensive T2 weighted abnormality on MRI compared to T1 GAD. So connecting all that together, there's, again, compelling data for supporting sex differences in this disease. Now let's connect it to response to treatment. So if you think about that example that I showed you a little while ago of when you look at the surgical, the surgical target is that T1 GAD abnormality, and then you have that T2 edematous, diffusely invaded extent beyond it. If you look in those regions, you find that a gross total resection means removal of that T1 GAD abnormality, the frank tumor region. And a subtotal resection means we're going to remove only part of that frank bright abnormality on the T1 GAD. So if there are more tumor cells invaded in the T2 abnormality for men than women, then you're leaving more cells behind no matter what surgery you're doing for men versus women. And that's basically what this slide says. Women benefit from even a subtotal resection. Even though those lines are subtle, there is still a statistically significant difference in improvement in survival, even with a smaller resection for women compared to men. In men, a subtotal resection doesn't receive a benefit. You actually have to get all the way to a sort of a gross total resection to capture some sort of surgical benefit from this disease. And I mentioned the chemotherapy that is typically used is temozolomide, and it's an alkylating agent. And you can track in the adjuvant setting with increasing cycles of temozolomide, sort of slightly different patterns of response, such that women sort of respond and they kind of keep responding such that their velocity of growth keeps going down and down and down. Whereas men, they sort of have this initial response and they plateau and maybe even come back up. And it turns out the rate at which they respond matters differently, such that if you have a big response for women, then that's really good. And if you have a big response for men, it's maybe okay, possibly because of this sort of recuperation behavior that they typically manifest. And another way to view that same data is to look at numbers of how many cycles of temozolomide a patient receives. And what you find is that it takes a lot more cycles of temozolomide for men to benefit than women. Women are benefiting at a smaller number of cycles of the therapy. Again, what's notable here is these are the standard of care therapies, right? So there's clearly personalized differences between the sexes in terms of understanding which patients are going to receive which benefit from therapies. So women receive benefit from a smaller surgery, and they receive benefit from fewer cycles of temozolomide. And again, that's the standard of care therapy. Hence the reason that their survival is on average a little bit more. So
Kristin, can I briefly ask then, Does that mean that potentially you're suggesting there's a different treatment strategy for men and women in the book, in the works?
Absolutely, yes.
Cool.
So one piece of the story is one of the reasons temozolomide is a useful drug and has shown itself to be the standard of care in this disease is because it has low to moderate blood-brain barrier penetration. So one of the challenges in brain cancer is that the normal blood-brain barrier out here protects the brain from drugs getting in, most drugs. There are limited chemotherapies that actually get in out here, okay? It has always been classically thought that regions here where the contrast is getting in. So if you're injecting these patients with a gadolinium contrast and it's leaking out, well, then where it's leaking out, the sort of classic dogma has been, well, if it's leaking out, then I'm assuming some drug that I'm giving this patient is going to leak out there too, right? So one of the, we have a PSOC U54 with MIT and Mayo Rochester that's focused on exactly this question of sort of, if you want to personalize therapy, you need to understand the differences in the biology between the sexes. That's that first component, hence the sex comma component. The second part is that we have to understand where the drug is getting in or if the drug is getting into the brain. And so that's what the second phase is. So to characterize this just a little bit more, so the blood-brain barrier affects drug distribution, as I mentioned. It's mostly intact throughout the normal brain, but it is porous in regions of aggressive tumor. What that means is where the gadolinium is leaking into the brain, and therefore you see it bright on the MRI. Those are regions where you presume something is leaking in, but we don't have a ton of data to support that. So how can we be sure if and where the drug is being distributed is sort of the premise underlying this U54. This is where I'm going to show you a little bit of data from that. So again, reorienting you to, if you've got this nodular tumor that doesn't have a lot of diffuse invasion. So here are very few tumor cells peripheral to the imaging abnormality. Here's the edge of what would be a gross total resection in black. And whereas the bottom patient has a tumor that's diffuse, has a lot of invaded tumor cells peripheral to the imaging abnormality. Again, gadolinium doesn't really penetrate the blood-brain barrier, right? So it's only going to leak into regions where the blood-brain barrier is broken down. And that's what this picture represents. So where this bright abnormality is where the gadolinium is getting in. And where this bright edge here is where the gadolinium is getting in. And in the middle is central necrosis. So the T2 weighted edema or swelling that I was referring to earlier around this abnormality is where you find those diffusely invaded tumor cells, as kind of visualized here by this mathematical modeling simulation. So if you've got a drug that's here and it looks like this, you expect sort of drugs on this end of the scale to look kind of where this imaging abnormality is. And so this is the regions of the tumor that's getting the most drug if the drug looks like this in terms of blood-brain barrier permeability. If the drug looks like this, where it's just highly permeable, then presumably it's going to get everywhere. Temozolomide is here, so that's a little tricky, right? It's kind of in the middle. So to draw this out a little bit more, imagine you're sitting in the center of this tumor and you're drawing a radial curve out. You draw a radial curve out, and this is the sort of tumor cell density you experience. That's just basically a radial map of this. And if you juxtapose a blood-brain barrier penetrant versus impenitrant drug, these are the patterns you'd see. You would expect, was our baseline hypothesis for this grant. Red, meaning where its blood-brain barrier is intact and functioning normally, then you would get lots of drug. And then you would get sort of decreasingly amounts of drug as you got towards the center, because the neovasculature in the tumor is, probably not the best vasculature in the world. It's going to be a little chaotic, and it's going to maybe not be as structured as well as the nicely, beautifully perfused normal brain. Whereas impenetrant would have a sort of a different pattern. You'd have this sort of opposite pattern where you'd have most of the drug getting in where this is breaking down. And you might have some diffusion phenomena where you're somehow getting some drug here in the T2 region, but then out here, you're getting basically nothing. So under this premise, we have done a series of both animal experiments and then human phase 0 clinical trials to be able to sort of build out the story. And what we find is this is looking at patterns of drug distribution for patient-derived xenografts. So these are patient-derived cell lines receiving specific drugs, and then looking at and then imaging those animals. This is not the imaging for them. This is human imaging, just to keep the orientation simple. But what you find is where the gadolinium has leaked in, there's a little bit more drug. where the gadolinium, where in the penumbra around the T2 weighted edematous region, there's a little bit of drug, and there's basically none out in the brain, which is consistent for a drug, a classic drug called erlotinib, which targets EGFR, which is blood-brain barrier and penetrant with maybe a 5% blood-brain barrier penetrance. On the other end, pick a drug, osimertinib, and osimertinib gets in, seems to be fine in the brain, and gets in similarly fine in the gadolinium enhancement where these leaky vessels are. But interestingly, there's more drug than you expect out here in this T2 edematous penumbra. So why is there extra drug in the T2? So we started building using these PDX models. This is the actual imaging of these small animals. You see the small MRI, and we spatially resolved the drug distribution using MALDI mass spec with a collaborator, Nat Agar at Dana-Farber. And when you overlay where the gadolinium is getting in, where the T2 weighted abnormality is, and compare that to the drug distribution, you could start using sort of machine learning tools to try and bridge across these scales. And what was notable is we expected gadolinium to be the big signal, yet T2 was the big signal. So basically, no matter what kind of model you're looking at, whether you're looking at drugs that are low blood-brain barrier penetrant or high blood-brain barrier penetration, T2 came out more often than T1 GAD. So this whole dogma about T1 GAD telling you where drug is going doesn't seem to be the case. Okay, so now let's scale up to the human. In the human, I referred to earlier this idea of image localized biopsies where you collect biopsies at different locations. Well, standardly in surgeries, when patients come in for brain surgery, they typically get two sort of types of drugs routinely. One is an antibiotic and one is an anti-epileptic. So levetiracetam is Keppra. It's an anti-epileptic, hence the reason it's highly blood-brain barrier penetrant, because it's working on brain cells and therefore it should get in the brain. And cefazolin is the antibiotic that's typically used, and that has low blood-brain barrier penetrance. And so we could use these sort of drugs as sort of controls and compare the drug distribution from where we collect on MRI with the peripheral blood drug distribution, and kind of sort of build a pharmacokinetic model in combination with a spatially resolved model of where on MRI is the drug getting in. And you see something not dissimilar to what I just showed you in much higher resolution on the patient-derived xenografts, is that where the leaky vasculature is, you get the most drug in both situations. But there's less in the T2 for the impenetrant drug, but the penetrant drug gets plenty of drug into the T2 window as well. And so that is, again, a reiteration that T2 is more important than we might have expected. And this sort of non-specific edema story is probably not, is probably more, there's probably a lot more to it physiologically than had been previously expected. So that's sort of the thread that we're following in that component of our work. And so what we really want to do is say, given you've got a patient and you know what sex the patient is, and you could say, okay, well then, given those imaging features, this is good or bad for outcomes, or this is a good or bad relationship to be able to predict optimal treatment for this patient. Which drugs should we use in our arsenal? Should we match with the imaging features? And so that's the kind of premise that we're sort of moving towards. So again, here's our two example patients. One is a not very invasive tumor, and one's a very, so we call that nodular, and one's a very diffuse invasive tumor. And if you look at these different therapeutic interventions, if you're do resection, patients like this with low residual disease afterwards are going to do pretty well, right? And so you'd think the maximal benefit would be patients up in this top corner versus this bottom corner. Whereas radiation, it turns out the more proliferative tumor cells respond more to radiation. There's an anti-angiogenic drug, Avastin, that bevacizumab, that drug works best for fast, big, quickly growing tumors. But drugs that fall into the category of either low or high blood-brain barrier penetrance, you would see different patterns, right? So you need to be able to sort out and match your categorization of each of these pieces to where that patient lives in the broad space of heterogeneity in terms of dynamics of the disease. So the first sort of drug that we were interested in looking at in the human scale was temozolomide, because everybody's receiving that drug. But as I mentioned before, it's somewhere between low and high penetrance. So it's kind of smack dab in the middle. So what is the result? The prediction here would be that there would be more benefit if you were more diffuse, right? If you're highly blood-brain barrier penetrant, and less, and in prediction here would be that would be more benefit if you're not particularly diffuse. And so most of your drug is getting here. That is temozolomide is probably falls on this end of the spectrum because it's only about 30% blood-brain barrier penetrance. So we'll surmise that. And basically that's what we found. Basically tumors with diffuse patterns and more invaded tumor cells, we had fewer cells exposed to sufficient amounts of drug, and those patients didn't do very well. Whereas patients over here that had fewer invaded tumor cells, and therefore gadolinium enhancement was where the most of the tumor cells were, those patients did fairly well and received a significant benefit from temozolomide chemotherapy. So all of this has connected patient heterogeneity in terms of at least sex. That builds bridges across the MRI techniques and how that connects to the biology underlying it and the outcomes. So it's a lot. Then in the middle, we have what about drugs? And the drugs are all over the place because they all do different things. They all have different blood-brain barrier penetrance. And everything up to this point has assumed that if you put drug in, it will hit target and target will be destroyed. But that's not the reality either. So glioblastoma is widely heterogeneous even within the tumor. And that intratumoral heterogeneity can be represented particularly challengingly because, yeah?
Oh, can I ask a question? Go back, like back to the sex differences. So, so the androgen signaling is a great difference between male and female, and androgen also is immunosuppressive, and it declines by age in male. Do you think like age, androgen, and microglia in the brain would add to those sex differences, especially that macrophage are also affected by hormones, at least in our data in prostate cancer?
Yeah, so first comment is my gardener is here, so things might get complicated very quickly. Statement one. Statement two is you're absolutely right, Asma. We are doing a ton of work in the space of microglia, macrophages, and GAMs, glioma-associated microglia and macrophages in the brain. Its role in their heterogeneous role in sex differences, and their heterogeneous role with age in both males and females. Mm-hmm. Thinking post-androgen, so post-menopausal women versus young women and men. There's a whole story there. There's a lot more. Have me come back in a year and I'll tell you all about what we're doing. And I would love to hear about what kind of things you're doing in this space, because it's a super interesting and super challenging. I'm really excited about it. So I would love to have presented about that today, but I didn't.
Oh, okay. Thank you.
We might have to, like, I don't know, this might get complicated really fast. So Gardner's coming. Okay. All right. So please warn me if you can't hear it all. So what about intratumoral heterogeneity? So, you know, when you have drugs that are targeted, you have to know if target's present, right? And the world of personalized medicine is all about targeted therapies, but we often, in case of GBM, we're often in the position that we have so few drugs to choose from for these patients that we give them targeted therapies even if we don't know if the target's present. So it's a complicated scenario. So when you see a failed clinical trial, it may or may not be having to do with whether the target was present or if there was no response because of things like blood-brain barrier permeance. So it's complicated, right? So what I mean by that is, you know, the classic approach to, hang on, just 2 seconds. I'm gonna try and convince him to stop blowing. All right, this might not work. I don't know, we'll see. Okay, so what types of cells are in the brain? So the classic one size fits all approach is that you go in, you biopsy a patient, you get the answer, and the answer is red. And so red, I've got a drug that targets red. Okay, great, personalized medicine, we're gonna win. And that's the sort of overall strategy, which has to do with the fact that we're limited in our ability to collect a lot of tissue in brain cancer. That said, the reality is that the brain cancer is a complex ecosystem of not just red and blues, but probably a rainbow. And worse yet, it evolves over time. So it's a dynamic, complex ecosystem that is not just reds and blues of politics, but in fact, shades of the rainbow. And so that complexity makes it particularly challenging to tackle this disease when you're in the world of personalized medicine, when we really don't have a lot of other things in our arsenal. Okay, so to tackle that, we have taken the strategy of, again, these image localized biopsies. So I'm going to reconstruct, and this is a patient that came to us with these first imaging on the left here. And this patient had, I think, a 17-day window between this first image and by the time they came to us to then go emergently to the OR. So this is an outside image, and you'll see the extent of abnormality on T1 GAD and the extent of abnormality on T2 weighted MRI. But what you'll find in just a moment is this tumor in this very short 17-day window progressed rather significantly. So I'll show you, hopefully that's 17 days later, here we are. That is a much bigger lesion than it was before. And at the time of surgery, we collected image localized biopsies at different locations within the tumor. And you see yellow are annotating where the biopsies were collected from. So those biopsies allow us to give a window into the biological heterogeneity of the disease. And we have a large protocol that focuses on capturing this sort of biological heterogeneity in the disease. Now with that data, we can apply machine learning. We can also use mechanistic mathematical modeling, but machine learning in particular, to build a bridge between the multi-parametric MRIs and the heterogeneity we see across those biopsies, to build maps of things like, I don't know, microglia and macrophages, T cells or tumor cells or proliferating cells, or this list could go on. And so we've built a series of these and published on a series of these that we use in a variety of different ways. So circling back to the sex difference comment, if you're just trying to look at using MRI features to predict EGFR amplification, or MRI features to predict PDGFR amplification, or MRI features to predict any one of these abnormalities, the MRI features that correlate with that amplification are different between the sexes. So again, reiterating and circling back to the idea that what you see in MRI means something different biologically for women and men. So much so that when you build these machine learning models with the combined data, you get some sort of accuracy like this. But in the individual data, your accuracy actually goes up, particularly for women. In this example here, goes up if you train only on female-only data versus male-only data, even though it's less data, because there's just simply a conflation of signals that are not the same. So skewed data at all, male versus female data, will skew your results for correlations between MRI and underlying biology. But how do you apply this? So applying this to regional understanding of heterogeneity in the disease, we go back to our PatientView database where we have tons of patients, and we go, okay, let's find some patient that received some targeted therapy and they had some interesting response. So here's a patient that had erlotinib chemotherapy, which is an EGFR targeted drug. And then here's their 1 month and 3 month follow-up, and you see regions of regression. That region is getting smaller. And then you see regions of progression circled in yellow. And on the top here is the T1 gadolinium enhanced T1 weighted MRI, and on the bottom is the T2 weighted MRI. And what you notice is why is this heterogeneity present? And there's lots of potential reasons. One is drug delivery, but another is intratumoral heterogeneity. So when you use those machine learning radiomics models that I was showing you just a few minutes ago, using those image localized biopsies to predict where is the tumor EGFR amplified, and you overlay that on this map, you find the region where there was response was the region predicted to be EGFR amplified, and the region for progression was a region that was not predicted to be EGFR amplified. And so now you can start building a bridge connecting all of those pieces of the puzzle to really get down to a personalized medicine strategy for individual patients. So I will show you a couple other examples. This is from a series of NCCTG clinical trials that targeted EGFR amplification. And I'm just going to hide the data and show this patient example got bigger. So the black region I kind of outlined in red. This is the tumor region before treatment, and this is the tumor region after treatment, after the first cycle. Here's one that was kind of stable, and here's one that was a responder. And we couldn't all these patients were put on the clinical trial. All of these patients were thought to be relevant to the clinical trial. They met the criteria. The clinical trial targeted EGFR amplification, and it was only the patient on the right that had a huge amount of that target present, and therefore had this response. The patient on the left that had no response didn't really have any target present. And so it was all very consistent that we could have known beforehand if we'd focused on patients like this, we'd have had a better response in the overall clinical trial. But when we conflate patients like this with this, we lose our signal. And so throwing out, I classically call this, throwing out baby with the bathwater when you're looking at these clinical trials. So, and to end on the sort of topic of the day is the world of immunotherapies. The immunotherapy makes this story even more complex, right? So we are in the world of both targeted targeted therapies, but also in the world of targeted immunotherapies. And immunotherapies are really complex from an imaging standpoint. And so this would be a typical pattern you'd see in a patient. So this is prior to this is a recurrent disease, prior to the plan to start therapy, and then this is the progression image, and basically here's their starting of an immunotherapy. And you see continual imaging growth. So the imaging is getting worse and then stabilizes and then starts getting better. So there's been no change in therapy in this window. This entire window, the patient's receiving the same therapy, there's no change in therapy. So this growth, this extension of the abnormality on MRI has been thought to be associated with some sort of immune inflammatory response. And as a result of that, the standard criteria for understanding when you change course in a patient with receiving immunotherapy, at least a GBM, is you're supposed to wait 6 months. But the median survival for this patient at day 0 here with recurrent GBM is 4 to 6 months. So if you're supposed to wait 6 months before you make a choice about changing therapy for a disease that only has a median survival at this time point of 4 to 6 months, there's an incongruity. So only patients that ultimately respond are the ones that are going to be able to last out this window. So imaging is not giving us what we need in this situation, and we can do better. So one of the machine radiomics models approaches that we've tackled has been looking at particularly T cells. And in this case, we're looking at patients on a dendritic cell vaccine and asking whether there's any sort of CD3 is a sort of umbrella marker for T cells. And in that, whether there are changes in patterns of T cell expansion as a function of treatment. So this is patients prior to the start of their dendritic cell vaccine. And this is the first imaging follow-up time point. So this is only 2 months in to treatment. And you see that there is some patients that decrease, and this patient didn't live very long. And then there's some patients that increase, and this patient lived quite a long time. And in general, on that vaccine trial, we find this general pattern that increasing abundance of CD3 cells predicted by imaging, even at the first imaging time point. So again, this is only 2 months, but difference between these pre and post imaging time points, identifies patients that are responders and that are going to do well. And then the patients that are decreasing, where their immune system has kind of like lost the battle and they're just leaving the game, then those patients don't do as well. So taken together, this sort of all fits into the broad paradigm of being able to have predictive tools that can not only tell you what the disease is going to do in an untreated context, but be able to identify patients that are going to best respond to individual treatments along the way. So in this case, this is some sort of mechanistic model simulation of not only surgery using that reaction diffusion system, but also radiation therapy for these patients. And being able to predict that proactively in individual patients is sort of the holy grail of where our lab is focused. And that is, I want to thank everybody that pays the bills and thank everybody that does all the work. Any questions?
Thanks, Kristin. I think that there were a few people on asking questions on the chat, and I think this is a good chance for them to start asking the questions publicly. I know that Asma asked at least one of them. But I know other people have, like Bryce. So does anyone have questions right now?
Hey, Kristin. I have a question.
Jill,
hey.
Yeah,
hi.
Hey, Jill.
Hey. Nice stuff you're doing. I'm interested in the pictures that you were showing with the EGFR. Those images, how were those created From the machine learning?
Yes, those are machine learning predictions from the sort of spatially resolved radiomics. So not radiomics that says that tumor has EGFR, but that says that tumor there has EGFR. So that's combining the image localized biopsies with the imaging.
Yeah, really cool.
Yeah, we have a CSBC UO1 that's focused on looking at like applying these in serial evolution over time. So there's a lot of, and trying to stitch that together using mechanistic modeling to understand the dynamics of different molecularly distinct subpopulations within the tumor.
Nice, thanks.
I think that Kathleen Wilkie has a question as well.
Hi, yes. Hi, Kristin. Thank you so much for an amazing talk. I was just wondering, with regards to your last stuff on the immune system, have you seen sex differences there?
Yes, lots. A whole nother talk. I could spend at least an hour talking about those. I have a graduate student that's particularly focused in this area.
Cool. So when are you going to talk about it?
Probably in the next, like if I had had slides, I would have been able to predict it like now, but we've got a couple things that are about submission. We've gotten several grants under review. If we get grant funded for this, then we'll be able to talk about it way sooner because then we can actually do more of it. But yeah, it's all burgeoning stuff. But I'm happy to share anytime, and I can walk you through like what we've been doing and what we're seeing. There's a lot of cool stuff coming out of it, but I just don't have it all synthesized.
Thank you.
I think Andre has a question too.
Hi, very, very cool stuff. It's quite amazing, these differences and stuff. But I'm sort of trying to understand the underlying biology behind these differences in the MRI images. Have you tried to correlate these to more kind of conventional histology images? Obviously, this dynamic data wouldn't be available, but just like to see, okay, what does it mean that you have less cell density in females compared to males? Is there a difference in the extracellular matrix, or the cells size different? Are you looking into sort of try to gain some biological understanding to figure out what might be going on to impact those observed fairly dramatic differences?
I feel like you might be a grant reviewer or something, because that's definitely the topics of most of our work right now. All the proposals that we have under review are looking at those basic mechanisms, like what is making this different? Why is the E2 bright on this both, but there's fewer cells? That makes what it kind of says is there's a different immunological response, and that immunological response has to do with expression of things like edema. So we have I have a junior faculty in the group, Pam Jackson, who's an associate director for my lab, and she does MRI biophysics work, and her MRI biophysics work is all built on like biophysical modeling of MRI physics to be able to extract the signal of things like edema from the underlying biology, to be able to drill that down more than a correlative way, right? More mechanistic-y, and building that bridge. But I think that is exactly where we're going, right? Is asking all those questions. And I don't know that I have an answer. I think we have lots of ideas of what the potential answers will be. But immune system, I'm sure, is going to be there somewhere.
Could you share like briefly some of these ideas? I'm just very curious.
Yeah, no, I think there's a differential threshold for inflammatory response in women and men. I think that's the foundational difference, which is consistent with broader biology, right? I think there's just a different threshold, and that different threshold manifests on imaging, at least that simple story on T2 weighted MRI. I think that's the key difference. And I think that's the brief summary of it. I think that's the signal.
Okay, very interesting. Thank you.
Hey, Kristin, it's Heiko. Nice to see you.
Hey, Heiko. Hi, nice to see you too.
Hey, I've got a question to the very early part of your talk, when you talked about the multi-modal and the early velocity in the MRIs as a predictor for outcome for male versus female. And you showed that the early velocity is more important for male than for female. Were those early velocities? Somebody's feedback.
That might be Chris.
But did you do? Was it the same early velocity for male and female, or did you do them separately by group?
Say that last part one more time.
So when you consider early velocity, is it the same velocity you apply to male and female, or does everybody have their own early velocity?
Ah, so is the threshold the same for each? Is that
Right.
what you're saying? Yes. So in that analysis, I think the threshold was the same. I think we took the median of the overall. I think that's what we did. We have several different versions of that result that basically say the same thing, right? But I think that's right, because I think that's why there's a skew towards there's fewer in one than the other pile.
So I just wonder, because there's so many things different, as you've nicely demonstrated, that if you were to normalize it for each individual group, if the results would be similar.
Yeah, one of the things that I think would be a message that I would implore everyone here, if you're looking at sex difference, don't add sex as just a biological variable in terms of like your Cox proportional hazard. Don't just say sex is one of your multivariate, because I don't think that's the right way to think about the problem. I think you analyze in males and you analyze in females, kind of to Heiko's perspective, because if the foundational pieces are not stitched together in the same way, adding as a covariate is not the right baseline assumption. And I think that is, if you get anything from today, I implore you to think of them as completely separate piles and see what happens.
Thanks, Kristin. Awesome stuff as always.
Thanks.
So I know that Bryce is trying to ask this question. I don't think he has the access to it, but I'll ask it for him. Given that tumors often perforate the BBB, is ctDNA a viable means of gathering data on tumor heterogeneity?
ctVNA, like CT, CTA, so CT art.
Circulating, I guess.
Yeah, so CTA, sort of. There definitely is a role, and it's not classically collected. The the data that we're more focused on, that's kind of, I think, analogous to what he's trying to get at, is the perfusion MRI, which is based on both. There's a couple different versions of perfusion MRI, but the net result of those is you get a relative cerebral blood volume. And turns out there are sex differences there too, just so you know. And that relative cerebral blood volume can give you some indication. So relative cerebral blood volume is a pixel-wise metric of fraction of a voxel that is vascular, is the idea. But there are parameters in that pharmacokinetic model that comes from that perfusion analysis that are K1, K2 sort of parameters like you'd get in a pharmacokinetic model of drug delivery in and out. And so those parameters are something we're also looking at because there is this likely difference in terms of how the brain vasculature responds to injury in the form of tumor.
Cool. Asma?
Okay, so since we saw the 6 differences mainly in invasion or the invasive end, do you think like there is another 6 differences in the progression of non-malignant brain tumors such as meningioma, where the invasive end is not what drive progression?
Yeah, so in meningiomas is one of the classic ones that things like hormonal dependencies are extremely important. So I would characterize sex differences in at least 2 different piles. One is the hormonal regulation, and the other is the intrinsic wiring of the pathways, right? The biological. Those are distinct. One is like sort of, I would just, and again, second element is implore you to keep those 2 separate pieces, because not all of this can be explained by hormones, all of what we're showing here. Meningiomas is a classic case in which there is significant changes and growth patterns as a function of pregnancy in particular. So women that are pregnant, my understanding is that meningiomas grow significantly faster during pregnancy than not. So there are known hormonal drivers. I do not know the basic biology of sex differences in terms of non-hormonal, but still sex-based differences between them.
Cool. I think maybe Etienne now.
I didn't hear who.
Etienne is
Etienne.
asking to be given the chance to ask a question.
Sure.
If he remembers to unmute himself, I guess.
I think he's unmuted.
Oh, okay. In which case, I mean, if not Sandy, I think you mentioned you have a few questions.
Yeah, so beautiful talk, Kristin, as always. Lots of cool new stuff, bridging scales that intrigues me, of course. And I have a question specifically related to that. But before I get to that, let me ask the other question. The other question is, to me, what you're showing is that there's some fundamental differences between men and women at the physiological level, at the tissue level, at how many levels we don't know. Is there a deeper understanding of those differences just in the normal tissue setting, right, in the normal conditions? Obviously, hormones is one of those, but is there others? And I remember having a beer with Josh, and one of the things he brought up was the whole sort of fight or flight kind of idea difference between males and females. But does that sort of understanding help shape what you're seeing in the data?
Yeah, if you could review grants for me, that would be awesome, but I don't think I can elaborate too much for that. So I don't think that's like a reality, but it does sound like you're listening to things that we're sending in. So physiologically, I think that's a particularly key element of metabolism, energy metabolism in particular. There are known sex differences in the baseline functioning and how shunting towards different energy metabolism based structures. So in fact, Josh Rubin, their AACR meeting yesterday had a virtual session on sex differences, and Josh gave a nice presentation on metabolic differences between women and men. So I think that is a key component of this, of certainly some of the imaging features that we see, that there are some baseline metabolism differences. But the story that I always like to say when responding to this is the idea of the cyclical famine. So this is something Josh is 100% Josh Rubin told me this, but it stuck and it makes sense to me, and it thinks about the foundational like wiring differences between men and women. So in the settings in which cyclical famines happen, there are a normal number of female babies born per like pregnancy. Percent chance that a pregnancy will produce a baby an infant is a typical number. There's a normal number of female babies, but they're abnormally small, where there's abnormally few male babies, but they're typical size. So the premise underlying that has been flushed out in a more foundational way is that female cells, when growth constrained or receiving insult, et cetera, they pause and wait for an opportunity to grow. Whereas male cells will respond to that and sort of grow at the cost, will respond to whatever the insult is and grow at the cost of viability. Because it only, evolutionarily, that makes sense from a perspective. It only takes a few men to populate a lot of wombs, as disturbing as that sounds, but it's true, right? And so, but from an evolutionary strategy, that all makes sense. So just thinking of that strategy down to the individual cell perspective, then of course the cells need to be responding to insult signals differently. And I just think that's another piece of the puzzle that I find particularly informative.
Yeah, that's intriguing. So my other question was when you hinted at that you're trying to use modeling to connect some of the machine learning results and some of the imaging and stuff that's occurring potentially on very different scales. So as someone who's known for PDE models, do you see this as something that's going to be PDE driven, or is it a different type of approach?
Well, right now we have definitely pieces that are PDEs. So we have a couple candidate models for competition of, there's two molecularly distinct subpopulations that are uncommonly seen at the same location in tumors. And that we have radiomics models for each of those pieces. And we're working with Marcus Owen and a graduate student, Bethan Morris over at Nottingham, who's particularly interested in building out these PDE models. So she's got a preliminary PDE model, which is already on archive. And then we're waiting for these maps to be optimized to, we're convinced that they're doing what we think they're doing, and gluing that story together. So I think the anticipation is PDE in the long game, but in the short game, we're also doing cell line and PDX competition assays between these different lines with these key molecularly distinct populations, and using that to get the sort of kinetic curves of the relationships between those to feed into the ultimate PDEs, but then like maybe validate those on the dynamics over time in patients. That's kind of I think the overall circle of what we think we're going to do.
Yeah, so sort of bridging the scales experimentally in some sense.
Yeah.
Nice, thanks.
Okay. Well, thank you very much, Kristin.
Thank you guys. That was a lot of fun, except for it was a lot of me talking to myself.
Yeah. Well, we were definitely listening, so you can see that.
No, and you know, the classic, you know, you're talking to your own screen. It's nice, but you know, maybe your voice is grating on yourself or something. I don't know. But thank you very much for the invite, guys.
Yeah, thank you very much. Okay.
Thanks, Kristin.
Thanks, everyone. Thanks,
Bye.
Kristin. Bye.
Bye.
Thank you. Bye.